Okay, now for our third phase, oxidative phosphorylation. Oxidative phosphorylation is going to be occurring in the mitochondria, specifically along the inner membrane of the mitochondria. Remember from a previous chapter, the mitochondria has dual membranes, an inner and an outer membrane. The inner membrane was actually much larger, much longer than the outer. So you ended up with all those little folds that we called cristae. On the cristae, embedded within it are going to be integral proteins, peripheral and integral proteins that are going to be responsible for setting up the energy that's going to be required for the making of the ATP. Now, when I say setting up, specifically what it is an electron transport chain. It's going to use those excited high energy electrons that are being chaperoned, transported in by NAD and FADH. And it's going to pump hydrogens from the inner matrix to the space between the two membranes. It's going to set up, remember, what is referred to as chemiosmosis, a high level of hydrogens on one side of the membrane, extremely low on the other. You're pumping from low up to high. The high will then flow through some sort of a transport protein. That's going to result in the process of making ATP. The electron transport chain, by its name, is a series of proteins. As I said, integral and peripheral proteins that are going to bleed off the energy from that high energy electron that was supplied in glycolysis and supplied in the citric acid cycle. By bleeding off little bits at a time, instead of taking it all at once, a lot of that energy is lost due to the second law of thermodynamics. Bleed off a little, turns out you're going to lose a minuscule amount each time compared to if you took it all at once. So, the electron transport chain, also known as ETC, series of transporters. Transporters that are active transporters. They are pumping. Pumping from low to high concentrations of hydrogens. Except here, even though they're active transporters, they're not powered by ATP, they're powered by, as I said, an excited electron. What we have is NADH bringing in its hydrogen. NADH becomes NAD. The electron stripped off the hydrogen, and this first complex, complex 1, it's going to take a little bit of that energy, pump a hydrogen. There you go. Pump a hydrogen across the membrane, from the matrix to the inner membrane space. It's going to pass that still excited electron to complex 2. Complex 2 with cytochrome Q is going to shuttle it over to complex 3. 3 is going to take, transport over another hydrogen. Cytochrome C to 4, transport another. Here's where FAD comes into play. FADH doesn't supply it cytochrome 1, supplies it cytochrome 2. Its excited electrons also go on to 3. 3 via cytochrome C to 4. 4 to right there. Boom. Oxygen. The whole reason you have to breathe in. You don't breathe in, none of this happens. Oxidative phosphorylation shuts down. Oxidative phosphorylation shuts down. After a while, Krebs cycle shuts down. Glycolysis shuts down. You die. That's why they say oxygen is the final electron acceptor. and it's right here. This is the whole reason why. That's the final electron acceptor. The oxygen you breathe in combines with the electron and hydrogen to form water. You make your own water. Now, you look in the textbook. Textbook goes through all the different proteins and parts of the complexes. No, I don't expect you to know it at that level, okay? That's going to be for cell biology. What I expect you to understand, series of proteins, integral and peripheral, 4 complexes, 2 cytochromes, 2 specific Q and C, NAD, FADH, give up their excited electrons, and the energy is bled off and used to pump hydrogens. There you go. The electron transport chain in 4 sentences. So you go through and you look at, from the textbook, it talks about cytochrome B within the complex 2, taking the hydrogen from the FAD. Ubiquinone, also known as cytochrome Q to some people, or just Q, takes that excited electrons from the complex 1, takes it from 2, passes it on to 3. 3 takes that energy, bleeds off, pumps a hydrogen, passes it on to cytochrome C, which then takes it to 4, the final complex. Once 4 pumps, takes that now regular electron, passes it on to back to hydrogen, and combines it with oxygen. Done. Minimal waste due to second law of thermodynamics.